Okay, everyone, let me try to show you something I've drawn here on the Jamboard. This is my first time using this Jamboard, which I think is interesting. Let me see if this Jamboard can facilitate me in accomplishing my objectives. Okay, first I've drawn 4 circles here, and these will be the diagram of monocot root, dicot root, dicot stem, and monocot stem. I'm going to show you the typical arrangement of tissues in the monocot root, dicot root, and so on, one by one. I really hope this Jamboard would work well. Okay, so I'm choosing this, the highlighter. What is this? Oh, brush. I think let's try brush. Okay, first we need to, I'm going to use pen first. Pen. Okay, if we use pen, what will happen? And then I use black. Then let's try I'm here and here, and I have a okay, here we got epidermis. Can we make the size of the letter smaller? Let's see if we can do that. No, oh, okay. How about this? Okay, I think this is good. I think this is good. Epidermis, but the problem is this one. Okay, I'm going to use pen again, and then I'm continuing this there, and I'm continuing this. Oh no, it's bad. Epidermis. Okay, so instead I'm going to erase this first, that, and this. Whoa, it's not easy to control this without using mouse. There, hopefully it's okay. Now epidermis is out here. Okay, so there it is. Epidermis is the outermost part of these plants organs. Okay, or monocot root or stem. The epidermis is the outermost part, and then we have these smaller circle inside these 3 structures, 3 parts, and actually these smaller circles are, I'm using I think I'm going to still use black. These are okay, I'm going to go here and go here. Okay, so I'm going to copy this part. No, cannot. I'm going then to write down here endodermis, and I need to move this thing here, I think. Okay, endodermis. Can I move this? Yeah, I can move this. Endodermis, epidermis, endodermis, and I've shared with all of you that no plant's body is really hollow. The parts of plants, all of them, root, stem, and leaves, must be filled up with something, and that fill up is actually a kind of tissue called parenchyma. Okay, parenchyma, and the presence of endodermis will separate the parenchyma, the ground tissue, the filler, the one here in between the epidermis and endodermis. We got the parenchyma, and then in here inside the endodermis, if it is exist, we'll see. Okay, now due to the location difference, then the parenchyma found and between the epidermis and endodermis is called cortex. Let me see, cortex. Okay, so I got cortex here, cortex, and I'm moving it here, and I got another cortex over here, and I got another cortex over here. I'm drawing the line which pointing out the cortex. Okay, and the cortex. Okay, now let me show you more things. Like I said, parenchyma tissue, parenchyma here is called cortex in between the epidermis and endodermis. If we still find parenchyma inside the endodermis, then this one is called, let's use this, pith. Okay, pith. We got pith over here, and we got another pith in the dicot stem. There. Do we have pith over here in the dicot root inside the endodermis of dicot root? No, we don't have it. Okay, while here there is no cortex or pith because there is no like boundaries or separated parts. So we call this ground tissues. So I'm going to use this again, and I'm writing it down, ground tissue. Okay, which is a parenchyma. Okay, I'm going to move it to the center. Now let me now draw the vascular system. Okay, I'm trying to use brush. Hopefully this one is good. And for xylem, I'm going to use red, and for phloem, I'm going to use blue. Okay, so I've told you before, I've shared with you that my way of memorizing which one is which related to xylem and phloem is xylem is bigger, deeper, while phloem is smaller, outer. Xylem bigger, deeper, phloem smaller, outer. So in case of monocot root, I need to first remind you that the difference between the root and the stem, in the arrangement of the vascular bundles, vascular system, xylem and phloem, is that in root the xylem and phloem are separated. Okay, they are not in a bundle, while in stems xylem and phloem are arranged and they are bundled. Okay, so again, I'm going to use red for xylem, blue for phloem. So to draw this xylem and phloem here in the monocot root, I'm going to draw it like, let me try. Okay, is it good enough? Okay, I can accept this. And then let's draw the xylem. We got xylem. I want you to notice that the position of xylem is deeper. I'm trying to draw it deeper. Things like that. Is it good enough? Okay. Now it's the Dicot root. This is actually my favorite one. See. Okay, I would draw it like this. Since I use red, means I'm drawing xylem. And it's like this, like star. Okay, and where's the phloem? The phloem is located somewhere here. Okay. That's it. Okay. Blue for phloem and red for xylem. Reposition like that. Okay. For the stem, the xylem and phloem are in the form of vascular bundles. Vascular bundles is a pack. It's a package in which xylem and phloem are arranged, and as the root, xylem is deeper, phloem is outer. Okay. So for that, I'm going to need things like this, an oval thing. Okay, and I'm going to need several of it. I think I'm going to need 5. I'm going to move it here, 1 over here. There it is. Not really good, but I hope this is representative enough. So I'm going to colorize the parts. So xylem always in, xylem in, xylem in, xylem in, xylem in, deeper. Okay, and now phloem is outside, outer, phloem is outer, phloem, okay. Now as for this, let's select this. If we copy it, okay, we got the so I'm going to move this here. Okay, now we got our randomized ovals here in the monocot stem. Let's try to colorize it so that it can be differentiated. blue for phloem, then it's supposed to be located outside. Blue for phloem outside. Blue for phloem outside. Blue for phloem outside. Phloem smaller outer. Phloem smaller outer. Phloem smaller outer. While xylem bigger, deeper, bigger, deeper, bigger, deeper, bigger, deeper. Now I already got the vascular tissues. Okay, so I'm going to now select this one and copy it, because here in Dicot stem we also got pith. Okay, now I'm going to copy this. There. So here we are. Here's what we got. So we got the monocot root over here, Dicot root, a monocot stem and Dicot stem, and this is the diagram. Wait, got something, almost forgot something. Epidermis is also here. There. Okay, this is it.